Morning, everyone. Good evening for me. Yeah, first of all, thank you so much for inviting me to your amazing symposium. It's a real honor to be one of the speakers amongst many really amazing scientists. Um, and it's an honor to start off this symposium. So, my name is David Van Dijk. And our lab is both at the med school and in computer science. So we build algorithms to decode the rules of life. And we are interested in all kinds of biomedical data. We specialize in large scale biomedical data. But we have a particular interest in single cell genomic data. And that's what I'm going to talk about today. Um, so, um, I would like to start by talking about single cell analysis in the age of large language models. So, what I would like to argue today is that biology has a language. It has maybe multiple languages, and we can use algorithms to understand and to decipher this language. So one good example of this is the immune system. So as you all probably know, cells communicate one another, and they use cytokines or chemokines to send signals to one another. So we can think of cytokines and chemokines as sort of the words. They're kind of the units of information. But these words or these signals don't occur by themselves. They occur in concert. There's usually many of them at the same time. In fact, they form sort of combinatorial signals. We can think of these combinatorial signals as sentences. So if we were to understand this language, the language of how cells talk to one another, this would be really useful because it would allow us to listen to cells and even speak to cells. So deciphering this language is important because it allows us to have control over biology. So the first project that I'm going to talk about is an algorithm that we developed recently, and it was published at Nature Methods. This algorithm is called CinemaT. And in this project, we set out to learn immune languages using causal inference. So let me start off by a question. So imagine you were to encounter an uncontacted tribe. So no one ever, talked to this tribe. They have a language that is completely unknown. They don't speak your language. You don't speak their language, but you want to communicate with them. So how would you go about doing this? Well, one way to do that, probably what you would do is you would start by listening. You would see, do they make any sort of sounds that I recognize? Are there certain patterns? And then you might try to interact with them. You might, give some gestures, show some gestures, or maybe use some symbol words or sounds and see how they react. You would sort of make guesses in how you could talk to them, and you would observe their response, their outcomes. And in the process, you would sort of refine and adapt your response and gradually build a model of some sort of language. And hopefully in the process, you would try to, or you start to be able to communicate with them. So this is actually what we're doing in biology. In some sense, biology has been uncontacted for billions of years, and only very recently have humans started contacting biology, namely through science, through biology, through experiments. So we can apply these same principles that we would apply to, when we would, talk to an uncontacted tribe or learn a new language to decode cell communication. And we do this using data. We make measurements. We perturb the system. And then using that data, we build models to try to understand the system and in some sense, understand that language. So one way to think about this is that talking to cells is or one way to talk to cells is by perturbing them, whether you're stimulating them with cytokines or perturbing them with drugs. You send them signal, you send them information, and you observe the response of the cells. And in the process, you might learn their language, or you might learn something about their biology. However, there is a fundamental challenge in understanding how cells respond to perturbations. In particular, when we're dealing with single cell data, with individual cells. The fundamental problem that we're dealing with is that measurement in biology is destructive. So, of course, there are some live cell imaging techniques, but any kind of high dimensional measurement, for example, single cell RNA sequencing, is fundamentally destructive. This means that we can only measure a cell once. And as such, if we want to understand how cells respond to perturbation, we have a problem. Because in an ideal world, we would measure a cell before we perturb it. Then we perturb the cell, whether it's with a drug or a gene knockout or cytokine stimulation. And then we measure the cell again, such that we can infer how the cell has changed. Again, the problem is we can only measure the cell once. So in this project, we set out to solve this problem using a causal inference framework. Specifically, we make use of what is called the potential outcome framework. In this framework, the goal is to infer counterfactuals. So a counterfactual is like asking, what would have happened if? Because matching cells between two conditions, even if you can't measure them in both conditions, but you can hypothetically sort of infer what a cell looks like in another condition, then it allows you to make causal inferences. So the way we do this specifically is we match cells based on so-called confounder signals, and I'll tell you more about that in a minute. We identify counterfactual cell pairs, which allows us to do true single-cell causal analysis. So let's talk more about confounder signals. So in this framework, we assume that the total signals, all the biological variability that is present in the cells, can be divided into two components, namely confounder signals and outcomes. Outcomes are all the signals that correlate with the experiment, with the perturbation. So a drug perturbation or a gene knockout, whatever you're measuring, any signal that correlates, that changes in response of the perturbation, we call outcomes. Any other signal, we call confounders. So you can think of confounders, or some examples of confounders is, for example, there's different cells, there's cell types, there is cell cycle. So there's all kinds of signals that introduce or that can explain variability in your data. However, they don't correlate with the outcome. So then what we do is we temporarily remove the outcome signals, and then we match cells between two conditions based on the confounder signals. And specifically, we use optimal transport to match distributions between the two conditions. So briefly, this is what our algorithm looks like. We start out with two cell populations, two single cell measurements. So for example, this can be control population and a treatment, for example, a drug perturbation. We perform single cell sequencing on these two populations, and we get our traditional cell by gene count matrix for our two conditions. Then what we do is we perform independent component analysis to separate the data into different components. We then identify the outcome component, so any component that correlates with the treatment, and any other component we call the confounders. Then we perform optimal transport, so we match cells based on just the confounder signal, so we temporarily remove the outcomes. So in theory, when you match on the confounders, you're dealing with two identical distributions, and this allows you to do correct matching. And we match the cells using optimal transport. And then we have cell pairs between two conditions that we can analyze. And this is what we call individual treatment effect analysis. For every cell pair, we can now ask, how did that cell change across conditions? So to do that, we bring back the outcome signals now that we've done the matching. So what do we obtain with this analysis? We obtain our counterfactual cell pairs. So these reflect hypothetical cell pairs as if it was the same identical cell measured in two different conditions. And now we can ask a unique question, namely, how does an individual cell respond to a stimulation? In addition to analyzing how individual cells or how cells respond to individual perturbations, we can also quantify, analyze how cells respond to combinatorial perturbation. In fact, we can do something that's very interesting, which we call synergy analysis. So we can measure cells in perturbation A, perturbation B, but also A and B combined. And then we can ask the question, how does perturbation A and B combined, to what extent is that different from just adding up the effects of the individual perturbations? If there's no difference, then there's no synergy, there's no interaction. However, if there is a difference, then we're dealing with a synergistic effect or an interaction effect. And this can be positive or negative, meaning when you combine the two perturbations, you can have a greater effect than the individual components, or you can have a lesser effect. Okay, so now that we have this causal framework for doing true single cell perturbation analysis, as well as synergy analysis, I want to show you how we can actually apply this in a real setting. So in this experiment, we set out to understand what we call the language of cytokines. So this experiment was performed by my amazing collaborator, Jeff Ishizuka, who measured immune cells in response to different cytokine stimulations, both acutely, so 2-day culture, or chronically after 7 days. And he measured them with single cells. He measured both individual conditions, so individual cytokines, interferon beta, gamma, IL-6, TNF, etc., but he also measured combinations. And those will be interesting once we do synergy analysis. So what is unique about our Cinema-OT analysis is that instead of showing single cell phenotypes, our approach shows single cell responses. So to visualize what I mean by that is on the left, we have our traditional single cell UMAP plot. Every point is a cell, and you can see that cells cluster by their cell type. So there's nothing special about that. On the right, we can see the same data, or actually a subset of the data. In this case, this is the interferon beta treatment. But every point here represents a response. In fact, it represents a counterfactual cell pair. So a cell pair that we inferred using Cinema-OT. What we're plotting here is the difference between the two parts of the pair. So it's the change. So interestingly, what you can see here, that even though there are many different cell types, as you can see on the left, there are only two clusters in terms of their response. There are only two ways in which cells can respond. And you can see here that actually one cluster of response comprises CD4, CD8 T cells and NK cells, and the other cluster represents monocytes. So you can see here that monocytes represent a distinct cluster in terms of their response. As I mentioned before, another unique thing that we can do is a synergy analysis. We can ask, how do combinatorial perturbations affect cells? Do cells respond in a special way to combinations? We perform this analysis, and using our framework, we were able to quantify the synergy score. So the synergy score is defined as the difference between the combined perturbation and the two individual perturbations. And as you can see here, there is one cell type that stands out as having especially large synergistic response, namely the monocytes. On the right, we zoom in on the monocytes, and we can see here that, for example, interferon beta plus IL-6 has a very strong synergistic response, because just interferon beta alone or IL-6 does not have a very strong response. So there's something special going on when you combine interferon beta and IL-6. So what does this show? This shows that our CinemaT method is able to quantify responses of individual cells to perturbations, but also it shows that it can quantify sort of combinatorial responses, and it reveals a combinatorial logic, if you will, that occurs in this cytokine language. So this shows that there are nonlinear interactions between cytokines, which in some sense you can think of it as forming the foundation of a language. A language is very nonlinear and it's highly synergistic. So the meaning of a word highly depends on its context. In the same sense, the response to a cytokine highly depends on the context or other cytokines that are present. Okay. Now I would like to move on to another project, which is also about biological language, which is the theme of this talk. So in the previous project, we kind of showed that there is some kind of language nonlinear interactions going on in this domain of cell communication through cytokines. So if we think that there is a biological language, or there are biological languages, what if we used state-of-the-art tools, state-of-the-art natural language processing tools to tackle this problem? And that's what we did in this project, where we teach large language models the language of biology. So we all are aware of a revolution happening currently in the field of large language models. So large language models are completely revolutionizing the way we interact with information. So in this project, we asked the questions, well, if large language models are really good at modeling natural language or human language, and we hypothesize and we have evidence for the fact that there is a biology, that biology has a language, could large language models understand biology? Can we teach large language models the language of biology? So how would we approach modeling language with biological language? Well, we can think of genes as words. So genes are kind of the units of biology in some sense. We can think of them as words. We can then think of cells as sentences. In cells, many genes come together, or gene expression patterns, determining the phenotype, the behavior of a cell. So we can think of that as a sentence. And then perhaps we can think of tissues or organs as paragraphs or documents. So this way, perhaps we can make an analogy with language. So why would we actually want to use large language models to model single-cell data? Well, first of all, large language models that are pre-trained on natural language and human language, they are inherently powerful. In fact, large language models can be seen as intelligent, systems. So they're already really smart. In addition, if we were to be able to use LLMs to model biology, it would allow for seamless integration of biological data, of sort of genomic data, and textual data, human language data. And finally, there's a rapid advancement of technology and infrastructure around LLMs. Every week there's a new model that comes out with more parameters. There's new techniques to do inference faster, to train faster, to have larger context, to fit more tokens in your memory, etc. So this is a really fast development. In fact, this development goes at an exponential pace. So if we were able to leverage these developments, that obviously would be amazing, and it would also be very hard to kind of compete with that if you build sort of a model from scratch. So one way to think about this is that instead of building a model from scratch, which is the traditional approach, right? You have a unique problem in biology, you come up with a new model. In this case, we already have a model, namely the large language model, and we don't mess with it. Instead, this problem becomes a data engineering problem where we transform the data into a textual format such that we can train LLMs on it. So we don't build new models, we do data engineering. And that's the philosophy of this project. So our approach is called cell-to-sentence. And cell-to-sentence is not just one computational method. It's a framework that redefines single-cell analysis. And the way the framework works is that first we collect a large amount of genomic data, single-cell data, millions of cells, different cell types, tissues, diseases, different perturbations. But we also include diverse annotation in the form of natural language. For example, papers and abstracts associated with the data, but also various gene set annotations, such as gene sets that we can include in this model. Then we train the model, or we convert all this data into a textual format, and I'll tell you more about that later. We then train our models sort of in a pre-training phase on this data. And then at inference, we have diverse downstream tasks, such as predicting cell labels, annotating cells, predicting perturbation responses, even answering questions about the data or embedding cells. So at the core of cell-to-sentence is a technique in which we represent cells as text. What we do is every cell is a rank-ordered list of gene names, ordered by decreasing expression level. So we simply take the gene names and we order them by expression level in decreasing order. And this is what we call our cell sentences. So this allows us to convert genomic data or gene expression data into a textual format. And it also allows us to, in an implicit way, encode gene expression data, because the rank represents the expression of the genes. So we take our single cell data, we convert it into a textual format, we train models, we do inference, and then we can go back to gene expression if we want to. So it turns out that this rank transformation is actually really efficient. We lose very little information. Here on the left, you can see that expression on the x-axis is highly correlated to rank on the y-axis, and we use very little information. On the right, you can see original data and reconstructed data, and you can see that they almost perfectly overlap. So we lose very little information by doing this rank transformation. So this is an overview of the different processes that happen in our framework. So we have our cell by gene expression data. We generate cell sentences by doing rank ordering of the genes. We can then add various textual annotation, for example, information on the cell types or disease or tissue, etc. We then train our models, our LLMs, on this data. This is standard autoregressive next token prediction. There's nothing special about this. Of course, in this case, it's next gene prediction, if you will. And then we have our trained models, and we can do inference. We can do downstream tasks. For example, we can generate cells from text. We can generate text from cells, or we can just do gene completion, predicting the next gene. And whenever we have generated new cells, so new cell sentences, we can even convert them back into gene expression. As I mentioned before, we lose very little information. And then you can work with the generated cells in your traditional standard gene expression space. So one way to think about a framework is that it reimagines single cell tasks as prompting. So prompting as you're used to when you work with ChatGPT. So for example, you can say, okay, generate the genes for a certain cell type, and it generates a cell sentence. Or the other way around. You give it a cell and you ask, what cell type is this cell? Or you can give it a bunch of cells and say, can you tell me about the biology that's going on in this data? So any kind of task becomes a textual prompt. And we have done this, we have tested this and benchmarked this. So here we generate cells from text. So we give it a cell type, for example, in this case, CD4 T cell melanoma, and then we ask the model to complete that sentence. And completing in this case means generating the genes for that cell in decreasing expression order. And you can see here that we have the original data, and on the right we have the ground truth data. On the right we have the generated data, and they highly overlap. We have extensively benchmarked this to compare this to state-of-the-art generative models as well as single-cell foundation models, and we outperform them. We can also do the reverse. We can generate text from cells. In this case, we gave it a cell sentence and then predict the cell type in text. Again, we extensively benchmark this and compare this to various sort of classification techniques, both sort of more general classifiers as well as specialized single cell models. Interestingly, we find that natural language pre-training significantly boosts performance. What I mean by that is that a model that's been pre-trained on natural language and then fine-tuned on cell sentences performs much better than a model that's never seen human language before. So natural language or human language significantly helps understanding biological language. And I find this really fascinating. And what that suggests to me that there are sort of general principles in human and biological language. There are similarities, even though you'd think, obviously, they're very different in how we speak and how cells speak, if you will. But apparently, there are common patterns because the model is able to exploit them. So, continuing, on this theme of, natural language or language of biology, we asked, can we go directly from gene expression data to biological insights? In some sense, we set out to automate the bioinformatician. I'm sure there are bioinformaticians and computational biologists in the audience. I myself am a computational biologist. And what we do essentially is we turn data into insights. In fact, we try to turn data into papers, right? That's the goal. We have data, we analyze the data, we write up a paper, try to get it published. That's kind of the process. So here we wondered, can we have an LLM do this? Can we train an LLM to go directly from gene expression data to natural language insights? And the answer is yes, it can. So the way we approach this is we train the model on pairs of data sets, gene expression data sets, single cell data sets, and abstracts, natural language abstracts from the papers associated with the data. And we trained the model to go from the data to the text. Then at inference, we gave it a new data set, so completely new data set that the model's never seen before, and we asked it, now generate textual insights. And it's able to do this. For example, this is one example. So this is our C2S model. and it correctly identifies the important biological sort of patterns or signals going on with the data. We wanted to kind of benchmark this, which is kind of difficult because no one has ever done this before. But basically what we did is we provided the same cell sentence to ChatGPT and Mixtral. So these are models that were not fine-tuned on cell sentences. You can imagine that they could still provide some insights because they might recognize some of the gene names and, be able to do something. And they were, but really poorly. So this shows that our model can actually learn to go from data to text. So in other words, it can translate, if you will, between biological language and human language. We quantified this, and then we greatly outperform language models that have not been fine-tuned on biological data. Another interesting application of our approach is that it provides a multi-species embedding. In fact, our model doesn't care about what species your data is. We trained our model on both mouse and human data. We gave it no information on how mouse and human relate. So we gave it no sort of information on homologous genes or other kinds of mappings between similar cell types or tissues, etc. We gave nothing like that. We just trained it on human cells and mouse cells, no extra information. Because our model is a language model, it can provide sentence embeddings, and this is a very common, widely used technique for embedding text or sentences, where you can extract a latent vector for a sentence. In our case, a sentence is a cell. It's a cell sentence. So every point here is a cell. And on the left, you can see that we embedded mouse and human data in the same embedding, because even though they're different species, we can map them to the same latent space. And they don't overlap, and they shouldn't overlap because mouse and human are not the same. However, you can see that they sort of have a similar shape. Now, if you look at the specific cell types that are present in this data, so this is a mouse and human pancreas data set, you can see that actually the two embeddings have a similar organization. Next, what we did is we figured now that we have a shared sort of latent space, even though there are two species, we can think of translating to the two species as a matching problem. Specifically, we used optimal transport to match mouse to human, as you can see here on the right. And this shows that, for the most part, it correctly matches cell types between the species. So we would expect higher probabilities on a diagonal. And you can see that for many of the cell types, it correctly matches. So we gave it no information on how the two species, the cells between the species relate. It was able to figure it out all by itself. We can also use our model to simulate perturbations. So as I showed before, we can generate cells. So we can train the model to generate responses to perturbations. In fact, this is the same data as the first project I presented, our Cinema-T method. So this is our cytokine stimulation data. However, in this case, we're using it in a generated sense. So what we did is we trained the model on half of the perturbations to go from perturbation to predict the gene expression response to perturbation for half of the perturbations. And then we asked it to generate an inference, the other half, and it's able to correctly predict responses to completely unseen conditions. So you can see here in B, the generated data overlaps the ground truth data. In C, you can see one specific perturbation. And in D, you can see on the diagonal that, for the most part, it correctly predicts perturbations. We did a similar experiment on this mouse lymph node cytokine perturbation data. It was generated in the Near Cohen lab at the Broad. This was 100 plus cytokines in different cell types. And again, we train on part of the perturbations and then generalize to completely unseen perturbations. And it's able to do this highly accurately. So when we think about modeling language, we don't model, individual sentences one at a time. when you use large language models, you generally, model a paragraph or a document. You could even, generate a whole book. It's always multiple sentences. And the reason for that is that the meaning of a sentence changes depending on the context. You can have this exact same sentence existing in two different books where it has a completely different meaning. In the same sense, in the same way, cells exist in a context. You have many cells in a sample, whether it's tissue or an organ or whatever your sample is, and the meaning or the phenotype or the function of a cell changes. It depends on the context. Because we are dealing with a large language model here, it gives us a really easy way, a really convenient way to model multicellular contexts. So we have built the first multicellular context foundation model. So instead of modeling individual cells at a time, we load multiple cells from the same context into the prompts, if you will, and then do inference. So we build models with 3 or 5 cells. And in this case, we predict cell types and tissues. And you can see here that the multi-cell models outperform the one-cell models. And when you think about it, this is kind of obvious, right? Because when you have multiple cells from, let's say, some tissue, it gives you a lot more information. And when you have one cell, you have more information to make a better prediction. So on the one hand, this is obvious. On the other hand, no one has ever done this, and we're the first to build this multi-cell foundation model. Okay. So to summarize, I hope I convince you that biology has a language or multiple languages, or at the very least, it's interesting to think about biology as sort of a language. So, cells can communicate through complex structured signals. Genes can act as words, cells as sentences, and tissues as paragraphs or documents. In the first project, Cinema OT, I showed that using causal inference, we can sort of uncover synergy as a fundamental building block of biological language, which revealed nonlinear interactions between cytokines. And that then presented about cell-to-sentence, which models cells as text, and then leverages large language models for single-cell analysis, which redefines single-cell research, allowing us to interpret cells, generate cells, predict perturbations, perform cross-species integration, and even model multicellular contexts. And with that, I would like to thank you. And I would like to thank the wonderful trainees in my lab who've done all this work and my amazing collaborators, as well as sources of funding. And I would love to answer your questions if you have any.